I'm so honored and thrilled to give this opportunity to share with you some of our recent work on tax mining. And I appreciate so much all the leadership vision and extraordinary work that have been accomplished by our colleagues at NLM. You guys are the ones paved the roads and build the bridges, and without these foundational platforms and tools, none of us could do our work. So thank you again, and thanks for the invitation. You can see that my appointments at UCLA are just growing every year with all these joint appointments that mandate me to go to faculty meetings, and then they decide that I'm not really good at attending the faculty meetings. And, and the appointments are just sort of piling on because the different schools and different departments want to highlight certain aspects of their programs. So I want to start this talk by dating back to a program that we were fortunately to be part of. And I saw Professor Andrew Su in the audience this morning. That was launched by NIH called BD2K. And it was really a couple of years before that when my lab first started transition from a wet lab with cardiovascular function disease and the role of proteins using proteomics tools gradually transferring to the lab of using data science tools and informatics. And in that journey, we were very much encouraged by our program officer, Dr. Susan Gregorick, to build alliance and collaboration with Professor Jiawei Han at that time at UIUC Computer Science, and Professor Wei Wang, Carol Watson, Hanningham Jacob from MOEBI at the UCLA-led HART BD2K team. And so the first time my lab ever looked at text data was the fall of 2014. Since that time, we have been growing and expanding, going full scale with proteomic data sets analysis in silicon PTM identifications, and a number of work and platform for enabling proteomics data sets AI ready, building metadata workflows for unique data types, for example, text data types, and more recently working with L on voice data types. And then using platforms and tools of LLM and generative AI supported annotation validation, and more recently, foundational models approach of federated learning on integrating single-cell omics and text data. I was alarmed this morning about keeping the talk together with the time limit. So I did prepare a total of 4 use cases. I'm only going to focus on talking about the first 2, and the last 2 each has only 2 PowerPoint slides because I have significantly truncated them to keep with time. We have made progress on some fronts and failed miserably on other fronts. The progress have always been welcomed by our community, and so we have had quite a few publications in different areas. The thing I want to say the most, in addition to the BD2K award, is the very generous support from NHLBI with 8-year support of R35, planning on full scale, that enabled my lab to transition from a functional wet lab to a lab now. I think 3/4 of us are dry labs now. The mandate of the R35, or if you look at the specific aims as listed in the abstract, are to provide a platform that could serve the cardiovascular community. And with that is our dedicated efforts to build cardiovascular-centric use cases and to serve the community. Many of us have built multiple use cases. If you Google how to create a use case, there are a number of suggestions and protocols. One that we happen to echo is this one listed by Indeed.com. It says, step 1, you need to know who are the communities where your users are coming from. So define user persona. Then you need to understand what are their goals. What are they trying to accomplish using your tools? Then you need to define the process, consider alternative outcomes, and compare the different use cases you built, and identify functionalities commonly shared or unique to each of it. And the last one of steps 6, it's very important because it says you need to repeat for all users. My interpretation of this step is to say user-specific use cases will bring you the specific outcomes that you wish to have, that it will bring specific functionalities that your tool and platforms can apply to, and eventually achieving sustainabilities of your tools and platforms. With that in mind, we have conducted surveys among 2 leading clinical and translational organizations of cardiovascular science. It's American Heart Association and the International Society of Heart Research. We ask questions of medical students, graduate students, postdoc fellows, clinical fellows, clinicians, and all investigators in the field. What is it that they're trying to achieve with proteomics or omics platforms or informatics platforms in cardiovascular translation and clinical science? There are different answers. I'm selecting the top few to share it with you, and you probably know this from day one already. We want the platform to help us achieve biomedical discovery, very tall order. Or we want it to be helpful in our hypothesis generation process. This is more general. We want it to help us to do knowledge synthesis. The clinicians say information and symptom indexing. We want it to help us achieve pathology understanding. And also among the top, but not the very top 3, is assist in disease prediction and clinical decision. And that may be a separate characteristic in cardiovascular biology and medicine versus cancer. What I have been informed by my colleagues in cancer is that apparently is their top goal in their user community. And then obviously there are other things follow, like drug discovery and things. So based on this information, we have formulated our study goal in the text mining investigations. we are to create user-centric workflows to perform domain-specific tasks on interpreting, understanding, as well as comprehending text data to achieve an efficient and effective and hopefully accurate way to enable biomedical discovery and clinical decision-making. And later I can share with you how well we've done with efficient and effective, and whether or not we're on track with getting to the accurate part of it. Going back to 2013 and the onsite of BD2K and collaboration with Jiawei Han, we have 3 publications in retrospect that have been meaningful for the progress we have made in the text mining direction. The first one was the first time that we collaborated with Jiawei to better understand the CaseOLAP platform Jiawei has built, and applying that to study a very focused, small, narrow, but important data sets in cardiovascular medicine, that's extracellular matrix proteins. We used CaseOLAP platforms to understand how proteins of this subproteome are relevant to the 8 major cardiovascular diseases that are out there, and how each of the disease is employing the extracellular matrix proteins as either part of the disease pathology or being modified during the treatment of the disease. The second report that was important early on is the creation and construction of a metadata platform focusing on text data. In this case, with Carol's leadership in cardiovascular medicine, we focused on rare diseases of cardiology and focusing on 3,300 clinical case reports that were able to collect and process from PubMed. I must emphasize that it was a particular study that both marked the beginning of an effort for text mining on clinician notes, because case reports are clinician notes, as using that for our later on work to dive into EHR. It was at the time that we recognized the significant challenges we were facing as a group, not a single investigator, to deal with the regulatory issues of accessing patient clinical note data from their physicians. Whereas we have this amazing resource on PubMed, over 2 million clinical case reports of multiple diseases ready to be shared and very much reviewed and ethically sourced text data sets. With that and building the platform, it started our ability to better understand the EHR data later. The third publication or reports that was important for us later on was the collaboration with Professor Wei Wang's group to build a cloud-based phrase mining and analysis platform using CaseOLAP to process text data and automate the process of entity recognition. At that stage, this was 4 years ago, we were able to accelerate the speed in this platform that were capable of processing millions of documents in a reasonable time frame, not 2 months, 3 months, 2 weeks, but rather few days. With those protocols in place, we began to use the platform to understand relationships among molecules and diseases, not a single molecule or small subproteome like the first paper we did on extracellular protein, but far more bigger list of proteins and molecules, not just genes or proteins, but also including molecular events and different processes. And so in this situation, I have to say, to scale took much longer than we originally anticipated. And that's one way where we learned many lessons to move forward. As promised, I will be sharing 2 primary use cases and very brief introduction of the other 2 to save time. I thought this is a good use case to start, is how we use the platform we've built to understand the role of oxidative stress, the molecules that initiate oxidative stress, the molecules that modulate oxidative stress, and the molecules that are outcomes of oxidative stress, how they contribute to the pathogenesis of the 8 category of cardiovascular disease. We selected this topic because a significant portion of cardiovascular studies and investigations will have the keyword of oxidative stress, because one or more molecules or molecular events of oxidative stress contribute to nearly all cardiovascular diseases that we see today. This project is led by Namuna, who's finishing up her postdoc fellowship in my lab, and Dibaka, who finished earlier, took a job as chief data scientist at Truveta, a Microsoft-sponsored startup company in Seattle, and Dr. Dean Wang, who's a project scientist in my lab. So our goal was to not to understand a single relationship, a list of proteins in a disease, but rather deciphering a number of proteins. These are calcium regulating proteins that related to oxidative stress and events, how they play into regulation, initiation, and outcome of oxidative events in the context of 8 cardiovascular disease categories. And obviously, you could appreciate when you have the relationship in different layers, the complexity actually adds the challenge of how you go about decoding them. So in this regard, you could appreciate the text data corpus, most of them come from PubMed, that are relevant to the 8 cardiovascular disease. There's currently about 1.2 million. And the 128 proteins using UniProt and Reactome leads to 128 not only proteins and isoforms, but nearly 500 reactome pathways. Whereas the most important part, the oxidative stress events of either initiating, modulating, and final outcome governing, has also 136 plus documents. We first mapped CRP disease relationships and assessed the disease oxidative stress relationships, and then determined the CRP as well as oxidative stress molecule relationships. Again, following the platform that was built by Dibaka a few years ago, together with Jiawei Han, we created 2 text cubes, one for the oxidative stress, one for the CVD, and then collected data of disease of interest, as well as oxidative stress events according to MASH from PubMed and UniProt, and began the entity dissection, entity recognition part, using case OLAP scores to add weight to the documents, and then using a number of analyses to dissect the differentiation and association. And this took advantage of the rapid development of graph-based AI networks. And so from very simple Neo4j aided the KG building to much more complex the GNN that we have applied, we were able to get the relationships out. So again, it went from document where these disease and molecules are represented to understanding the relationships of the molecules with the disease in the context of oxidative stress. Clearly, you could imagine there's a number of graphs, and the way the graphs are constructed, the key is it enabled us to go in using the prediction link to identify links that was previously not known. And in this sense, I can go here for another 2 hours to explain the data, but I want you to recognize for the first time, we are able to link relationships, very meaningful relationships, using very simple, straightforward algorithms that we download and then customized, like total neighbors, to identify critical molecular events that happening as the initiation of oxidative stress in a particular type of disease, and how they're being regulated by calcium regulating proteins. And to me, it's a job. If I have 5 postdocs or a team of grad students 10 years ago, it would took them 5 years to do. And now we're looking at it in a few days. And we can twist parameters and easily apply this platform to other questions that interest the cardiovascular community. So I'm going to speed up a little bit. So the next use case took place over a year ago, and we were excited that we not only had the understanding of the BERT platform by NLM, and also the recent support of the LLMs. This is a project, it's important for me to show this slide, led by a brilliant grad student who's about to graduate in a few months, Alexander Pelletier, Dr. to be, and a couple of other very able and enthusiastic partners. So what Alexander has done is he's created a platform, he calls it Retrieval Under Graph-Guided Explainable Disease Distinction. Very similar to cancer, the cardiovascular disease span 8 major categories and over 500 subcategories. To be able to accurately treat the disease, to prescribe therapeutics that lead to meaningful outcomes, really requires an in-depth understanding of the disease pathology and molecular events underlying the disease. Alexander created this platform to do jobs again that would take months, years of many grad students to do, but you can now to do it in a number of days or a couple of weeks. It has 4 sections. It downloads and accesses the data sets from either PubMed or any other knowledge bases of relevance, and then triage protocols to the text data sets you have and integrate them with data sets from other knowledge bases. And from that moment on, the text mining protocol we have would first identify the protein disease relationships and construct knowledge graph. The third section enables explainable AI to train the graph attention network, build relevance ranking analysis, conduct predictions, and then get to the last part where we use BERT-based or assisted summary process to generate output for knowledge synthesis. In this process, we use the system retrieval augmented generation that I saw many of the speakers report this morning. In our case, we applied entity indexed text data sets from together the knowledge graph, conducting user queries with the aid of 3 system agents. So specifically in our RAG part of the system, we extract, interrogate information from the knowledge graph, generate node and edge embeddings of molecules and disease, create a vector database, and then conduct similar search from name entity recognition and relation extraction. Most importantly, we create Cipher query agent interface with the user to conduct retrieval, validation, and refinement with the help of 2 more agents, query verification agent and reasoning agent. Furthermore, in the case of text data retrieval, we use entity relation documentation, query, user query again, and then text evaluator agent, very similar to the other agent in the knowledge graph and reasoning agent. From there on, with the help of BERT, we could do cascade questions of evaluation of a disease process. And if you ask any clinical fellows from cardiology, they'll tell you that's exactly what they want to do. So 1 question, 2nd question, 3rd question, all questions are linked and relevant to understand a disease. In this case, we have a cascade of 7 questions. The first 5 and 6 using knowledge graph information from the text data, and the last 2 using the AI predictive analysis. So I'm only giving 1 example. User, what are the currently prescribed drugs classified as beta blockers, arrhythmic drugs, and anti-fibrotic drugs? And then BERT aided Raj system answer right away. Which drug is the most commonly prescribed in each category? You will have the answer right away. If I'm interested in 2 of the 8 diseases, which you could change parameters at any given time, all drugs relevant to the treatment of the disease will come out. It will rank based on weighted predictions, and it will give you the molecular targets of these drugs. At the end, most importantly, it generates notes and letting you the path it took to lead to the outcomes and the answers. And I'm most proud that we're actually getting to something very meaningful. The example that I have here in the green arrow is to show atenolol is traditionally a drug for lowering blood pressure. And this is a strong prediction that it may be a good drug to treat arrhythmic cardiomyopathy. And it is a lead that many of my clinical colleagues are following right now, because the output we recommended and the specific PubMed publications that Bert helped us pointing to gave sufficient strong evidence that the leak is reasonably interesting in a real world scenario. So I don't have time to go through the next two. As I said, I have actually truncated them, but I want to say we appreciate the platforms that many of the pioneers have provided in the field, including a couple of you sitting in the audience, and with Jiawei and Wei. We think we definitely have done a lot with respect to create an efficient and effective way for text mining. With respect to accuracy, I think this is a challenge we need to focus on moving to the future. Thank you. I have a job to advocate for CFDE, and you may find the little pamphlets. It's a great pan-NIH program that's dealing with the things we're all interested in: data integration, data harmonization, and data fair and data AI ready. I also want to give acknowledgement to the program that I'm part of. It's the NIH Bridge to AI program. Thank you again.
Thank you. We do have a few minutes for questions. If there
okay.
are, yeah, we have until 3, so we've got a couple minutes. Do we have a question to start us off?
I was super clear.
All right.
Thank you for a very interesting talk. I was wondering, the example that you'd shared where you were using RAG and sort of an edge node model, how does that handle similarity for sort of non-identical edges? So can you capture things where it's not the identical relationship, but something that gets in sort of the same idea? How does this?
So in our specific case, because the 8 category of cardiovascular disease has, the mother descriptors of the MASH. And then there are many other subcategories of the disease, I think 147 and then over 500. So you do see the edges. I don't have that PowerPoint slide. You do see the edges come out as similarities. So the figure I had right here, this one, you could appreciate the disease with respect to similarity of edges. You see the point 2 and 3, they are all of them are off from the same MASH, mother descriptor, cardiomyopathy. And then the subtype as restrictive and dilated. But in this case, the system is able, through weighted protocols, to identify which of that is important. So the 2 and 3, you could say, definitely have different relationships. For example, 2 goes to 7, but 3 does not. Yeah. And the other thing I want a little bit too fast is the text mining part. So with clinical case reports is what we dealt with the most. You have abstracts. Sometimes it's very short, 2 or 3 sentences. And then you have the main body and you have the discussion. Contrary to what we have done years ago, in the recent work, we actually go in to differentiate the text in the abstract versus the text in the main body, because all of you probably noticed that the abstract contains major sentences for conclusions and reasonings and causes. And so those are the words have to bear more meaning or weight. And whereas the sentences somewhere just appear, it may or may not be constitute a real information there.
Thank you. Do we have any other questions? I think I'll go ahead and, well, please.
Thank you so much. I wanted to ask you to maybe comment on how much we can conclude from one positive example. So, you've presented this case that you've described as your colleagues wanting to pursue now and looking at based on the evidence that was provided. But do you have any concerns at all about whether that was a one-off or whether, the outputs of these systems are reliable enough for us to kind of proceed with this as a paradigm change?
So I think you raised the question of accuracy, and that is the component among the three. We're putting a lot of efforts working on it. And right now you probably saw the system response from the rocket. It's really based on BERT-based summarizer, gives specific PMIDs on the reports the evidence is drawn from. So it doesn't just come out as a black box to say, oh, based on this, I have this. No, it says, I have XYZ evidence. These evidence are from original articles. These evidence are from a review article. These evidence are from 3 clinical case reports. So what we're doing right now is saving time, or the clinical fellow will tell you, and saving life. So a cardiology fellow could look at this instead of going through 120,000 articles, read very carefully these 6 articles that are relevant to this, and draw a meaningful human in the loop final conclusion on what that is.
Thank you so much.